Hello, and welcome to the last part I believe 6 or 7 of Chapter 6 on microbial metabolism. Here we're just going to cover some of the basics of photosynthesis. In photosynthesis, we are converting light energy into chemical energy. Light energy is a form of radiant energy that is captured by structures within photosynthetic organisms called photosystems. And this diagram looks really kind of busy here, but I just kind of want to show you what's going on. Photosynthesis occurs in two reactions. We have what's called the light reaction. And the light reaction produces ATP and an energy intermediate known as NADPH, and uses that to drive or provides the energy for the dark reaction. Now, in the dark reaction, light is not a part of this, obviously. It's called the dark reaction. And the dark reaction is where we get glucose, which is a form of carbon fixation. Now, we're not going to go through all of the different steps of this, but I want you to know the difference between the light reaction and the dark reaction, the purpose of the light reaction, how it functions, and then how that is applied to the energy produced for the dark reaction. So here in this diagram, we have the light reaction, and the photosystems, I know it's a little confusing. Photosystem 2 does occur before photosystem 1. They are only named this way because of the order in which they were discovered. But actually, light energy electrons are initially brought to photosystem 2. And the electrons used in the light reaction is an electron transport chain. And the electrons for the electron transport chain in the light reaction comes from water. So hydrogen atoms are broken off of water molecules. The protons are used for being pumped across the membrane. This is all occurring in plants. This is occurring in the chloroplast. And it will pump these protons across a membrane, and that will create a proton motive force. Meanwhile, the electrons that are ripped off of those hydrogen atoms get put into these photosystems. Now, in the prokaryotic and eukaryotic electron transport chain in central metabolism, those electron chains rely directly on the energy from the electron itself. In photosynthesis, light energy is used to excite the electron beyond its natural energy state. So it actually gets the electrons more excited than they would normally be, causing them to release more energy than they normally would. As these electrons get moved from one photosystem to the other, they lose a little bit of energy, so more light energy is introduced to the second photosystem in this pathway to re-excite that electron. That electron will finally get attached to and will reduce NADP. NADP plus will become NADPH. NADP reductase is the enzyme that carries out that step. So the terminal electron acceptor in the light reaction of photosynthesis is NADP. Okay? And we make NADPH. But we've also created with photosynthesis, we've created a proton motive force. And that proton motive force will also use ATP synthase. And it works exactly like the other electron transport chain, and it produces ATP. So we'll get a bunch of ATP from this process as well. So in the light reaction, we make NADPH and ATP. Now, after the light reaction, the light reaction can occur in two different pathways. I'm going to show you a quick video here to show you the difference in electron flow that occurs in photosynthesis. This most often occurs in plants. However, sometimes the cell has enough ATP, or the cell is not using the dark reaction, or the cell does not need any more NADPH. In that case, it will change the flow of electrons, and sometimes the electrons will be in a cyclic flow to produce ATP only, and then other times they will go into a non-cyclic flow to produce both NADPH and ATP.
In bacterial photosynthesis, a single photosystem is involved. When an electron is energized by absorption of light, it is ejected from the photosystem reaction center. The electron then passes to ferredoxin and then down through the cytochrome B6F complex, plastocyanin PC, and finally back to the reaction center. The energy released during this electron transport generates a proton gradient which is used to produce ATP. Since the excited electron returns to the reaction center, this mechanism for making ATP is called cyclic photophosphorylation. The reducing power needed for biosynthesis is not generated in the process of cyclic photophosphorylation. Plants and cyanobacteria utilize two photosystems which work sequentially to produce both energy and reducing power. First, a photon of light ejects a high-energy electron from photosystem 2. The electron lost from photosystem 2 does not return to photosystem 2, but is replaced by an electron generated from the splitting of water and the production of oxygen. The electron then travels from the excited reaction center of photosystem 2 to plastoquinone, to the B6F complex, to plastocyanin, and finally to the reaction center of photosystem 1. This electron transport system generates a proton motive force that is used to produce ATP. Since the excited electron does not return to photosystem 2, this mechanism for making ATP is called non-cyclic photophosphorylation. When photosystem 1 absorbs a photon of light, it ejects a high-energy electron. The energy from this light absorption is used to generate reducing power in the form of NADPH. The ejected electron is replaced by an electron from photosystem 2. In bacterial
So in the light reaction, we produced some ATP and some NADPH. Now that ATP and NADPH is used for the Calvin-Benson cycle, also known as the dark reaction. And the reason for this is plants and photosynthetic organisms are taking inorganic carbon sources and converting them into organic carbon sources. They are doing this so that they can produce their own glucose molecules. Think about it. All a plant can do is absorb some nutrients from the soil, but it's not absorbing sugar, it's not absorbing glucose, but it's still using central metabolism for ATP production in its cellular processes. So these organisms are capable of taking inorganic carbon compounds like CO2 and jamming them together in a process known as carbon fixation. Now, the Calvin-Benson cycle and this carbon fixation process is really very complex. It's an incredibly complex process. So we're just going to look at it in terms of phases. So we have stage 1, stage 2, and stage 3. So in stage 1, we are bringing in, right, this is bringing in, and this is known as the carbon fixation stage. So we're getting a bunch of CO2 molecules, jamming them together to create 3-phosphoglycerase. From here, we move into stage 2, and in stage 2, we have more carbon fixation occurring, and this is also our oxidation stage. And in this particular stage, you can see NADPH that we produced in the light reaction is being used to produce NADP+, because we need these phosphate groups. Up here in this carbon fixation stage, look at this, it required 12 ATP. That's a lot of ATP. So that's why the light reaction devotes the majority of its ATP to the dark reaction. Now, right here at the end of stage 2, look at this. We get glyceraldehyde 3-phosphate, or G3P. That molecule pops up quite a bit. So G3P is produced, and the cells then take G3P and put it together like Lego blocks to build whatever molecules they need, including glucose. But not every single G3P that's produced in the Calvin-Benson cycle will be used. Some of them will move back into a whole bunch of very complex reactions to create new molecules using more ATP to start stage 1 again. So in the Calvin-Benson cycle in the dark reaction, ATP and NADPH from the light reaction is being used in a carbon fixation process to produce G3P molecules for the cell to build whatever it needs in its normal primary metabolic processes. Now, anabolic pathways. We've spent a lot of time talking about catabolism and the breakdown of molecules in order to derive electrons and energy from these molecules for cellular processes. But the cellular processes that this energy is driving are more often than not going to be anabolic. And so biosynthesis is necessary for a cell to function. Cells have to build proteins and enzymes, and they have to build all of the different machinery that makes it a cell. So in glycolysis, the different steps of the pathway, remember those, we call them precursor metabolites. Well, here, just as when lipids were broken down and glycerol entered here, it also can leave here to go become a lipid. So molecules, intermediates, can either move directly through metabolism and produce lots of ATP, or some of these intermediates will actually leave before complete breakdown and become precursor metabolites, or the beginning stages of a new molecule. We produce the amino acids cysteine, glycine, and serine from those start as intermediates in glycolysis. Same thing with phenylalanine, tryptophan, and tyrosine. They just come from a different intermediate in glycolysis. We get the fatty acid chains produced in lipids from acetyl-CoA. We get amino acids from the Krebs cycle. So different two areas in the Krebs cycle are intermediate precursor metabolites. Excuse me, I had to remember my term here. For bacteria, from the pentose phosphate pathway, two of the intermediates there are precursor metabolites for some amino acids that are used by those particular bacteria. So just as we put things in, we might have broken down lipids somewhere outside in the cell and taken those products and put them into central metabolism. We can also remove those products from central metabolism, and we can use them to produce. Now, what do we use these biosynthetic pathways for? We make amino acids, lipids, nucleic acids, peptidoglycan, and glutamate. So these are the 5 primary pathways or primary compounds that we are producing, that bacteria, I should say, prokaryotic cells are producing in biosynthesis from central metabolism. So just a quick review, make sure in photosynthesis you know what the light reaction versus the dark reaction is, what are the 2 forms of electron flow in the light reaction. There's another video in your textbook online that is very helpful on cyclic versus non-cyclic flow. We have circumstances, when would you use a cyclic flow and when would you use a I'm sorry, there's a typo here, this should be a non-cyclic light reaction. What are the different, what is anabolism, where in central metabolism do we see anabolic processes being used. So I hope you enjoyed our multi-part series on metabolism, and I will see you guys in the next lecture. Have a good one.